everyone, I'm Ella Shiratsky. With me here is Luis Valentin, and this is Metagenomics Lesson 4, Read Mapping and Using Bowtie 2. So let me share my screen. So today we're going to talk about read mapping. So for starters, we're going to talk about what is read mapping, how do you map reads, different options for read mapping tools. I'm going to briefly mention how Bowtie 2 works, or the algorithm behind it, and then we're going to do a tutorial on how to use Bowtie 2 and SAMtools to map reads to a reference genome. So let's start with what is read mapping. When you get your sequences back from Illumina, you get a lot of short reads. But you do have a reference database that you're interested in seeing whether those reads actually align to it. So what you're doing is you're taking the read and you're starting to, in theory, go over the genome position by position, saying, does the read match here? Does it match here? Does it match there? Does it match better there than here, etc. And you're doing this millions of times. So obviously they've had to develop quite complex algorithms to do this quickly. Now, why would you want to map reads at all? Reads give you information about whether a genome or an organism is present or not in a sample. It can tell you what is its coverage. So it's sort of a measure of relative abundance of the genome in a sample. You can tell if there are variants or different strains of this genome by using reads to look at single nucleotide variation. And then finally, you can also use the coverage that you calculated to bin MAGs, or metagenomic assembled genomes. So I talked about how you take the read and you start aligning it or matching it to different positions in the algorithm, in the genome, sorry. So how does this actually work? Very generally, like I said, you go position by position, and then you start comparing your read to the reference genome. And for each base, you're going to say, okay, does this match the same position in the genome? Is it a mismatch? So is it a different base? And then for a more complex alignment, is it even not there at all? Or do you need to insert a base to the reference in order for the read to be properly aligned? So each of these has sort of a price tag. If you have a match, then you get a reward. If you have a mismatch or an indel, then you get penalized. And when you do that along the entire read, you end up with a sum of all of these penalties and rewards. And that is your alignment score. Now, as you compare the reads to different positions in the reference genome, each one of them is going to have a score. And what you're going to want to do is pick the best score, the highest score, which has the least number of mismatches and indels. But of course, it's a little more complicated than that, and the highest number of matches. Now, there are a lot of different scoring matrices, which means they give a higher penalty, for example, for an extended gap. So when you have to insert more and more and more bases into the reference, that becomes more and more expensive. Or maybe they penalize mismatches more. So one of the most famous scoring matrices for proteins actually is called Blossom 62, but there are a lot of different ones. So in here you see basically how Blossom 62 works. If you have a match, then you get a positive score that's along the diagonal. And then for each amino acid, if it mismatches, like A and G in this case, it's not a big deal. For an A in your read and a P in your reference, you would get penalized -1. But if there's a W in the reference, you get penalized more. And that has to do with what are the odds that an amino acid would be substituted by a different one. But there are also scoring matrices for nucleotide sequences. So there are a lot of read mapping tools. They differ mostly in their scoring system. The most commonly used ones are BWA, or the Burrows-Wheeler alignment, Bowtie2, which is also based on the same algorithm, and BBMap, but there are many more. If you are interested in a comparison between popular mappers, I've added a link here to a blog by Maren, or Murat Eren. If you're unfamiliar with him, get into his blog. He's super helpful with anything bioinformatics. So he compared mapping with different aligners, and it's a really useful blog and very easy to read. Okay, so today we're going to focus on Bowtie2. And the way Bowtie2 works is, first you take your reference genome and you index it. What it means is you just turn it into a format that's easier or faster for the aligner to read. And it has to do with just rearranging the letters. So let's say an A would be 1, and a G would be 2, a C would be 3, and a T would be 4. So instead of saying GATC, you would say 2134, or 2143. So. And it's just faster for the aligner to read it that way. It doesn't matter, you're never actually going to look at the index yourself. It's just an input that you give the aligner. Once you've created your index, and that can be based on any FASTA file, then you start aligning your reads to that reference. There are basically two main types of alignment. It's either end-to-end, so you have to have the ends of the read aligned to the reference, which may mean that you need to enter some deletions in your reference, like on the left example. Or you could have a local alignment, which means you don't have to include all of the bases in the read, and then you might have some bases hanging off the edge, representing potentially insertions. There are pluses and minuses to each approach. Most of the aligners, I think, use end-to-end by default. Luis, do you agree?
Yes.
Cool. All right. So the way Bowtie works is it gives a score to the read depending on, again, mismatches and indels. And then it calculates sort of a mapping quality summary score for the entire read. And the score has to do not just with mismatches and indels, but also with the quality scores of the read itself. So for example, if you have a mismatch between a base in the read and a base in the reference, and this specific base has a very high quality score, so you're really sure that it was read correctly by the sequencer, then you can assume that this is a real mismatch representing maybe a variant or a strain or something. Whereas if the quality of this read in that position is poor, then it's actually more likely to be a mismatch due to sequencing error or amplification error, and not a real single nucleotide variant. So it's a pretty complicated algorithm, and fortunately Bowtie will do that for you. So there are two ways in which reads can align to your reference genome. Either they are concordant, meaning they are facing in the direction that you expect them to, and they're not too far on the reference genome or further than expected. Whereas you can have a discordant pair, and discordant pair means that either the reads are not in the orientation that you expect. So for example, they're both in the same orientation, as opposed to facing each other, or you could have a very large insertion in the middle between those reads, much bigger than, let's say, the insert size that you aimed for when you did your library prep. And Bowtie will recognize that automatically. Obviously, you might want to think about maybe throwing out your discordant pairs. There are not going to be tons of them, so you won't be losing a lot of data. So just something to think about. Once you align your reads, Bowtie will give you an alignment file that's going to be in a format called SAM, or the binary form of SAM, which is BAM, binary SAM. And this file, we're going to start by looking at SAM files, and they have a specific structure that gives you a lot of information about the alignment. So it'll tell you the Q name or the name of the query. That's the name of the read, like the actual header of that read in the FASTQ file. The reference name, so if you have different contigs or chromosomes in your reference, it's going to tell you which one it actually aligns to. The position, so the leftmost position where this read aligns to the reference, and that's the position in the reference. And then the MapQ score, so this is that summary score of how well the read aligned. And it's going to look something like this. So you're going to have, this is your read header. This is your reference name, so it's aligning to chromosome 20. The leftmost position where it starts aligning is 190,930. And then the MapQ score is 3, which is fine. There are a lot of interpretations of which MapQ score you should use. I think generally, my understanding is that more than 1 is fine. After that, it's going to have something called a cigar string. So you see this 100M here. A cigar string is a representation of how the read aligned. So M represents matches. So in this case, you just have 100 matches. So 100 bases in the read are matching the reference without any indels between them. Here is something that is probably more of a discordant read, because you have 50 matches, then 4,700 ends, so ambiguous spaces, which means there's an insertion into the reference, and then 50 matches. So this is not a great alignment. It's kind of weird, actually. Am I right about this?
It was just an example to show the different parts of the file, but yeah, I took that from Google, so it's not actually from one of my genomes.
Okay.
But you're right, yeah, it's not a good one.
All right, cool. So finally, before we get to the tutorial, today we're also going to mention how to deal with SAM and BAM files, which is really useful for downstream analysis. So we're going to talk about sorting your alignment file by the chromosome position on the genome. And you can kind of imagine why this would be useful, because then you can imagine calculating the coverage in a certain range on the genome, for example. So let's switch to tutorial. So here is our binder. Okay, so when we go into our data folder, we're going to be using these files. So we have, this is the reference genome, which is from a disulfobacter. And then we have, two samples of paired reads that we can map to it. So originally the reference genome came zipped. I unzipped it. You can do that easily. So you just type unzip and then copy paste the genome name. I already have it here, but I can tell it to replace. Just replace it. All right. So the reason is that Bowtie doesn't recognize the zip format. It only recognizes gzip. So I'm pretty sure that you can give it a gzipped reference, but not a zipped one. Okay, so the first thing that we're going to do is we're going to build that index that we talked about. And we're going to do that with Bowtie to build. And we're going to give it B. I forgot how to do this. Sorry. You just give it the reference genome and then the alias that you want to call it. So I'm going to call it DVH. And let's see what it does. Okay, that's it. That's all it took to build a reference or an index from one genome. And if I hit LS now, I have all these files that end with BT2 or Bowtie2, and these are my index files. I'm never going to refer to them specifically. I'm always just going to call the alias, which is DVH, and it's going to know to look at all of these files. So let's look at an alignment of just single end reads, because sometimes that's all you have. So we're gonna run Bowtie2, and the first parameter that we're going to give it is -x, and this is your index. And so all you're going to write here is the alias of the index, which is DVH. And then you can give it your single end reads with a -u. I'm pretty sure that the default is you don't even have to give it a -u, but for the sake of order, let's do that. And then that's the gzipped forward read file. And then S is your SAM file output name. So let's call it single example.sam. Let's see what happens. It's going to be pretty quick because it's just one genome and just one not very large FASTQ file. And that's it, it's done. We had 100,000 reads in this FASTQ files, 94% of them aligned properly. About 6,000 of them didn't align at all. So maybe they are, I don't know, there was a deletion in the genome and now they don't map to that, the part they map to doesn't exist anymore. About 91,000 aligned exactly in one time, and then about 3,000 aligned in more than one place on the genome, and that does happen. You can choose to ignore those files if you want, those reads if you want, but in this case, we're not going to. So let's look at our file, our SAM file. It starts with a header that tells you about your run parameters. And then it starts giving you information about the alignment of every read here. So this is the read name. And then it tells you, this is your cigar string. So it mapped with 151 matches, no mismatches, no indels. Then it's going to give you the sequence of the read right here and the quality scores of the read. So all of these weird random symbols, they represent the quality of the read, and that's because quality in FASTQ files is not in on a decimal base. It's represented by all sorts of symbols that represent numbers between 0 and 93, if I'm not mistaken. So each one of these really represents a number between 0 and 93. And that tells you what the quality of the reads are, or it tells Bowtie anyway. So this is what a SAM file looks like. Anything else we wanted to show here? This is the leftmost position on the genome that this read maps to. And this is going to be the MapQ score, right?
Yes, you're right.
Okay, cool. Awesome. Sorry, I keep calling on you, but
No, that's great. Yeah,
I'm not sure.
You can cut all these sections, and that's nice.
Awesome. All right. So this was mapping just one single read. Let's see how we map paired reads. So I'm going to do the same thing. Bowtie 2, give it the same index, but this time I'm going to tell it -1 is the forward read. So let's say we use this sample and then give the matching reverse read. So I'm just going to change this to 2 with the -2 flag. And once again, we have the SAMtools output file name. And this time I'm going to call it paired example.sam. And the other new thing that I'm going to introduce here is the ampersand and arrow. And that means that I am directing the output that would have gone on the standard output like this. So instead of writing it onto the screen, onto the terminal, I'm going to write it into a log file, basically. And if I only had the arrow, then it would only write the output. Sorry. If I add the ampersand before the arrow, it's also going to write any errors that during the run into the log file, if there were any errors. So let's call this paired example.log and run it. And again, this should be really, really quick. Probably 10 seconds or so. And then we can look at both the log file and the SAM file. Now, the reason to keep these log files is because you have all this information of how many reads actually map to your genome, which is information that you're later going to need when you write the paper. So make sure that you keep these log files. So you can have this, you don't have to recalculate all of this later on. All right, we're done. So once again, we got a SAM file. Let's look at the log file actually. So we can see if there were any errors when this ran. So it looks like no errors. And then Bowtie tells us, again, we had 10,000 pairs of reads. About 28,000 of them didn't align concordantly at all, which is a lot. It's a little surprising, but okay. About 70,000 aligned concordantly once, which is exactly the kind of result we would expect. We're mapping reads to a reference genome. And then another couple thousand aligned concordantly more than once. So of these weird 28,000 reads, about 20,000 aligned discordantly. So maybe the orientation wasn't as we expected, or the reads were too far apart.
Yeah. Also, you can trim the reads before you run Bowtie, and that will also clean up the data, like have some PCR errors.
These are not trimmed?
No.
Oh, okay, that explains it. All right, so I'm actually surprised that we're getting such high overall alignment rates.
Yeah, I just remember, I was like, wait a minute, that's really, oh yeah, I didn't trim the data.
That explains everything. Welcome to Bioinformatics troubleshooting. This is how it works.
Yeah, also that happened to me once. I was like, wait, what is this so bad? And someone told me, oh, do you trim the reads? Oh, no.
Yep. Your instructors are not infallible, and it does happen to us as well.
To us.
All right, cool. So to exit this view mode, I'm just going to hit Q, and here I am, or here we are, back to our data folder. Okay. So we've learned how to create our SAM files or the output files, but SAM files they tend to be pretty big. So if we look at the size of the SAM file, about 80K, sorry, is it, yeah, 80K. This tends to add up because we're only talking about 100,000 reads here. And when you have a lot of samples and every sample has millions of reads, these files get really, really big. You certainly do not want them on your personal machine, and you also don't want them hogging the server. So SAM has a binary format to it, which creates much smaller files. It's called BAM. And the way we convert a SAM file to a BAM file is with a suite of tools called SAMtools. So SAMtools. SAMtools has a really useful function called view. And what we're going to tell view is we want to convert to a BAM file or SAM file. So SAM to BAM is going to be written like this, -BS, capital S. And then we're going to give it our SAM file name, and direct the output to the same file name, but ending with BAM. So let's do that and see what happens. All right, cool. Now, we can look at the size of the BAM file just to compare. And it's about a third of the size. So that's pretty good. Another useful thing to do with SAM or BAM files is to sort them by their position on the chromosome that we talked about, which can be really, really useful in a lot of downstream analysis. So the way you do that is with a command called SAMtools sort. So you would take your, you can run this on BAM file. BAM file, and I'm going to direct it to a file called BAM, here an example, sort BAM. Now I expect this file to be the exact same size because it's the same file, it's just sorted differently. So let's just make sure. No, it's actually smaller. Why is it smaller? Okay, I don't know, it's smaller, whatever. If you wanted to run both of these commands at the same time, which is really useful, you could do it like this. So you have your, this is how you convert SAM to BAM. And then you would pipe that. So meaning you take the output of this and throw it into the next command. So pipe, and then SAMtools sort. And then you direct the output in here. So you could run multiple commands with this pipe as long as you keep, the pipe just keeps throwing the output of the previous command into the next command. So yeah, we can run this again just to make sure that we get the same file and it looks like it worked the same way. And then the, I guess the last command that we're gonna learn is how to index. So indexing a BAM file is kind of like indexing your reference genome. Just creates a file that's easier to read for certain software. So for example, later on, if you're gonna use Anvio, it's gonna take an index BAM file. The way you do this is with SAMtools index, and then all you have to do is give it the BAM file name, and that's it. It's super quick. And now, if I look at all of my files called pair example, and I'm gonna have the BAM file. Here's the BAM file that I just created, the sorted one. And this is the index, it's gonna end with BAI. You're never gonna look at that file yourself. It's just gonna be used by bioinformatics software later on. So even though BAM files are binary, so if I was looking at this BAM file just with head, why is this not working? Yes, obviously this is not legible in any possible way. But if I was using the SAMtools view command and I gave it this file, And then I piped the output of this into less, which is to start showing me the file. Now we can see what's actually written here. So this first column is the read name. And here we have the position on the chromosome. And as you can see, these reads are sorted by the position on the chromosome because we sorted this file. So yay, it worked. All right, so Q to exit this view. And that's about it. That's all we have for today. So thank you for joining our tutorial, and thank you to Luis for actually constructing this tutorial and helping out with the PowerPoint. And I wish he could actually just give you this tutorial himself. He'd probably do a better job.
No, come on. Yeah, let us know if you have any question or any comment or concerns.
Yes.